Okay, so good morning, everyone. I think I'm the last one of the Congress, so I wish you are not so tired to, to listen to me. I'm going to talk about a protocol that I'm currently developing to propagate alders, trees from the genus Alnus, on in vitro conditions to generate clonal lines of this tree. And why should I focus on this specific tree? Okay, I don't want to focus too much on this, but the other importance is mainly ecological because of the riverbank stabilization, it's a nitrogen fixing plant, and so many other things. And I also want to be brief about this because Raquel already talked about this yesterday, so. But we have a very important problem concerning almost all parts of Europe and also some parts of United States, that is the alder decline. Just to remember, it's caused by several species of Phytophthora, and so many trees are dying because of this disease. But how did all this happen? Like, how did we get surrounded by all these alder decline, all these Phytophthora? Because all Europe, as I said, or almost all Europe are affected by this disease. So the problem, as many other forest diseases, is the globalization and the climate change. The nurseries, the human movements, had allowed, and also the increase of the temperature, had allowed the Phytophthora species in this case to spread and to reach areas where they were not present. So the problem here is that in this case, the alder species and alder varieties were not used to this kind of pathogens. So the problem is they didn't develop defense mechanisms to face this disease. So a good approach to study this, to see which are the physiological mechanisms, the genetic mechanisms that can give the alders chances to tolerate the disease, could be the in vitro culture. With in vitro culture, you can generate clonal lines with a specific genotype, and for example, you can develop a clonal line that is that you can identify with a tolerant genotype, and another one with a susceptible genotype. So, of course, it already exists protocols to develop alders in vitro, but what I was trying to do was to find a way to produce in vitro alders, clonal alders, in a fast way and in an efficient way with a high rate of survival. So my first approach was to take directly the buds from the trees on the field and to cultivate them. Like normally, to avoid contaminations, what is normally done is to at first cultivate it ex vitro, and then sterilize the surface of the plant and do the in vitro culture and so on. But this is time consuming, and it has a very low rate of survival, success, because some of the branches that you try to propagate ex vitro usually die. So, of course, I had a problem that was a lot of contaminations. Of course, I sterilized them on the surface. I will explain it later because it's always the same protocol in my case. But I increased the amount of bleach, increased the amount of ethanol, and every time I had contaminations of Alternaria alternata, because I identified the fungus that is present in the environment, and even sometimes the boots die, but not the Alternaria, so that's a very big problem. So my second approach was to take seeds from the field and cultivate them to generate the source of explants. So the thing is that by this way I can I can germinate in axenic conditions the seeds, because I don't add sugar. That is you can say that is the source of contamination, because the seed has nutrients to the plant to grow. So the first step was to collect seeds from October to January, because it's the best. I found that was the best period to do so. I Berlinized them during 1 week at 4 degrees, and then I wash them overnight, and I treat them with gibberellic acid, which is a phytohormone. I'm going to talk a lot about phytohormones, that promotes, as you can see here, the germination of the seeds. This is a common mechanism. If you pay attention to this, it breaks the dormancy of the seed and increase the rate of germination. So once I do this treatment, I sterilize on the surface following this protocol, and then I put them in wet chamber with these conditions that will be the same for all the experiments in a cultivate chamber for plants. So once I have cultivated, I had the seeds germinated, I transfer it to a WPM medium, which is a common medium for woody plants, which had these compounds. It's just nutrients, so it's not worthy to mention them, and always with 7 grams per liter of agar. And that's what I have here, the seeds germinated. At this point, we don't have in vitro plants, because the in vitro plants, these are not clonal, and these are not from explants. These are F1 generation, but they are totally sterile. They don't have bacteria, neither fungi, so this could be a very good source of explant. Then I follow this protocol that is modified from an article that managed to cultivate to do in vitro culture, but with this kind of low rate and more time consuming, but it was very helpful, of course. And I reduced the amount of sucrose. Okay, the growth would be not that fast, but then we will manage to avoid more the contaminations. And then add that is a cytokinin that promotes the growth of the shoots, and a low amount of auxins. After that, again, with a low concentration of the sugar, in this case glucose, change the different kind of hormones to promote, again, the growth of the shoot, and not that much, but I was reducing the amount of cytokinins and maintaining the amount of auxins to promote slowly the growth of auxins. I forgot to mention that at this point I put parts of the tree of this seedling. So if you use just one seedling, okay, when it's more, it has a good length, you can have here a piece of it. Let's see, another piece of it. There are the explants with nodes and internodes, and these then you can have clonal lines, because this and this, of course, has the same genotype. And after that, I follow, as I told you, these cycles, and then the routing. I transfer to new recipients with more glucose. At this point, we assume that there's not fungi, and the half of the WPM, the half of the nutrients, and then you have in vitro culture. I'm currently in these steps. I have already done some plants, so I have a little amount of them. So this could be a good approach to study, to have clonal lines very fast, and to study. The next steps would be to study in genetic ways and also physiological ways. We can do a lot of things, like inoculate in vitro the Phytophthora and study a lot of things with these in vitro plants. So thank you all.